What does it take to be a true pioneer in medicine? Today we'll explore the evolution of medical courage at the Hebrew University, from the legendary researchers of a century ago to the modern scientists battling today's most persistent health challenges. And what better place to start than in Jerusalem of the 1930s, courtesy of S.Y. Agnon, the great Israeli novelist.
Now then, in this period, when Herbst was working out of doors, when he used to amble back and forth, strolling, pondering, thinking his thoughts, he ran into a friend, a fellow professor at the university who had just recovered from a serious illness. He was an epidemiologist who used to travel everywhere to study the course of contagious diseases. When the unrest in the world was such that he could no longer travel, he stayed in Jerusalem and worked at home. Now I will reveal in a whisper what was whispered to me. One day, he wanted to study a particularly deadly tropical disease. But in all of the land of Israel, he couldn't find anyone suffering from it. He exposed his own body to the disease and tried to cure himself with the drug he had invented. Great German doctors report in their memoirs that when they were trying to fathom the secret of a disease and its cure, they would expose one of their patients to it. Not this Jerusalem scientist. He tested the disease and the cure personally on his own body, and in doing so he almost died. Now that he was recovering, he often went out for walks. When Herbst first heard the story, tears rolled down his cheek. One day, he spotted the doctor in the park at the end of Rashba Street, at the very edge of Rehavia. He and Herbst were not closely connected. One of them worked in the humanities, the other worked in science. But since they worked in the same institution, they did know each other. When Herbst saw him, he bowed, kissed his hand, and went on his way.
That passage was from the novel Shira by the Israeli Nobel laureate S.Y. Agnon and translated by Ziva Shapiro. It's a work of fiction, but the scientist, who was so dedicated to his work that he used himself as a literal guinea pig, he was quite real. Today we'll meet Professor Saul Adler, one of the fathers of Israeli microbiology, and a pioneer whose daring spirit and groundbreaking work laid the foundation for the Hebrew University's global reputation. Adler and his colleagues fought epidemics that once threatened life in this region: malaria, leishmaniasis, diseases that shaped the fate of nations. But epidemics haven't gone away, they've evolved. Where malaria once dominated, today we confront antibiotic resistance, bacterial persistence, and even obesity. New threats requiring just as much ingenuity and courage. Welcome to The View from Mount Scopus, a podcast from the Hebrew University of Jerusalem. I'm Gilad Halpern. In this episode, we'll journey from the courageous leaps of the past to the cutting-edge discoveries of today, meeting scientists, perhaps slightly less unorthodox than Professor Adler, but no less dedicated or innovative. But let's begin in the beginning. Born in 1895 in Belarus and raised in England, Saul Adler came to Jerusalem in 1924 with a passion for Zionism and tropical diseases. The first world-class scientist recruited by the Hebrew University, he was the founder of the Institute of Microbiology, one of the university's 3 inaugural research institutes. Around the same time, another professor at the young university, Israel Kligler, was waging war against malaria. Adler and Kligler were colleagues, but couldn't be more different. As historian Daniel Gavron writes,
"The two men were a study in contrasts. Kligler was small, bald, self-assured, outspoken, even belligerent, and American. Adler was shy and ostensibly modest, a handsome, laid-back, charming Englishman. Both were brilliant researchers who would earn international reputations and leave their marks on the university and on Israeli science in general."
In those years, the Hebrew University was growing fast. A vibrant research culture was taking root, sowing the seeds of the robust scientific community we know today. Amid this expansion, Professor Adler was carving out his niche. He could be quite the absent-minded professor, as Israeli biophysicist and later the country's fourth president, Ephraim Katzir, recalled.
Adler's reputed absent-mindedness was confirmed by my brother Aharon, who was lucky enough to attend one of Adler's courses. Aharon and his friends would find him at his microscope in the laboratory, having forgotten about the lecture. Arriving a little out of breath, he would ask to be reminded where he had left off, and would then continue in great detail, without using any notes.
This was all intriguing enough that I set up a meeting with Daniel Gavron, the man who literally wrote the book on Adler, in his home in a village just outside Jerusalem.
I'm Daniel Gavron, Danny Gavron, but Daniel professionally in my writing. Why Adler? Well, it was by chance, but I became very interested in him, although he wasn't alive anymore. But a lot of people, his daughter-in-law and other people, were around. I mean, I can't say that, I was inspired by Adler, but as I wrote the book, I became more and more fascinated by him.
What was it that really made you fascinated by him?
Well, his character. He was a very, very, like myself, tended to be a bit left-wing. He was eccentric, he was unusual, he was absent-minded, and he did some very valuable work. I'd never heard of leishmaniasis, but apparently it's one of the 6 big killers in the world, and he did pioneering work on it. Adler himself was born in Belarus, today's Belarus, but grew up in England and became a doctor and went out World War I, went out to Iraq in the Middle East, and he was an army doctor there. He became interested in tropical medicine and went to the Liverpool School of Tropical Medicine. And at some point, he got a job in West Africa, but Chaim Weizmann, the first president of Israel, who founded the Hebrew University, knew about him as a fellow scientist and recruited him in 1925. He came to mandatory Palestine and founded the microbiology department, which was part of the Hebrew University.
Well, one of the 3 first institutes. What made him leave England and come to this godforsaken place?
It's interesting. He was a Zionist of a sort, but he grew up with a very Jewish background. He was terrifically well-sourced in Hebrew. He knew the Bible, the Talmud, the Mishnah, the Gemara. He was very, very good, and it was a perfect fit in a way, because he was a coming young scientist looking for new things to discover, to find out, to work on. But his Hebrew was fantastic. So he was one of the very few of the early Hebrew University people to staff who could lecture in perfect Hebrew. And in fact, he wrote a biography in Hebrew of Charles Darwin, which is regarded even today as a classic of modern Hebrew scientific writing.
Adler's research was groundbreaking. In the 1920s and 30s, it focused on leishmaniasis, a parasitic disease sometimes nicknamed Baghdad boil or Jericho rose. In British Mandate Palestine, it caused painful skin sores but was rarely fatal. Adler had encountered a more dangerous form of the disease during World War I in Mesopotamia, and he was determined to conquer it. The story goes that when he couldn't find local patients to study, Adler exposed himself to the parasite, nearly dying while testing a cure, just as in Agnon's tale. What was his own contribution to the study of leishmaniasis?
Well, he really, in his day, was the world's leading expert on the disease. It's a disease that's, in the Middle East, is just a nuisance. It creates all sorts of boils that turn into scars and that kind of thing. But it wasn't fatal. In India and China and places like that, it was a killer. And therefore, what he discovered here about the origins, for example, it was transmitted by sandflies and mosquitoes, and the various treatments for it were very significant. Not so much in Israel as for the Indians and the Chinese. I think he was very aware of the Israeli contribution to medicine in Asian and African countries.
Adler's fearless approach set the stage for decades of innovation in microbiology. But understanding microbes isn't just about fighting them. Sometimes it's about listening. Today, at the Hebrew University's Faculty of Medicine, Professor Sigal Ben Yehuda is doing just that, decoding the conversations that bacteria have among themselves.
Hi, my name is Sigal Ben Yehuda. I'm a researcher at the Faculty of Medicine of the Hebrew University, and I'm a microbiologist. Our lab studies the complexity of the bacterial social life. Basically, we're interested in understanding how bacteria communicate with each other. And our most exciting finding is that bacteria can communicate in a contact-dependent manner. Basically, they can form these very tiny bridges that we term nanotubes and physically connect one bacteria to the other.
These nanotube bridges between bacterial cells were invisible to science until Ben Yehuda and her team observed them a decade and a half ago. Imagine two bacteria sitting on the surface, extending microscopic tubules of membrane that reach out and touch, creating a direct channel from one cell to the other. Through these channels, bacteria can literally share parts of themselves. Professor Ben Yehuda and her team revealed this wordless molecular language of bacteria by watching fluorescent tagged molecules flow from cell to cell under the microscope. What they found was startling. Bacteria can function as a connected community. So it's a physical connection between the two.
Yes, it's a physical connection, and it exists mainly when the bacteria are stationary. They are like staying under sessile conditions, like they are on solid surfaces. So they produce this network of nanotubes, and through these nanotubes they can exchange molecules and even small tiny DNA plasmids.
How did you find out that they exist?
Basically by microscopy. So this is our specialty. So two types of microscopy. One is a high resolution electron microscopy, which we can visualize that under fixed conditions. And the more exciting one for us is the fluorescence microscopy, in which you can see the production of these nanotubes and their dynamics in living cells.
So until you came along, nobody knew that there was this kind of physical connection between bacteria.
So there are a few types of physical connection that have been described throughout the years, and the most prominent one is the conjugation machinery, but these connections are made of pilus and are proteinaceous. That means that they are made of proteins. And the junction that we found are different. They are made of membranes. That means that the cell membrane is protruding, is sending this extension. These extension are then engaging, like searching for the appropriate recipient cell. And then the connection is established.
So what's passing between the two bacteria through that connection?
So this is one of our major research questions, of course. But we know that small molecules, for example, like nutrients, amino acids, can be delivered and exchanged in between the bacteria. For example, if one bacterium is deficient in making one amino acid, it can donate the amino acid to another bacteria which is living next to it. Maybe even more strikingly and more importantly for us, they can exchange antibiotic-resistant genes, for example, and even antibiotic proteins. So they can provide each other with the even temporary or permanent ability to resist antibiotics. And how is that important for us? For example, if we have one bacterium which is resistant to antibiotic, but it's a very nice, non-harmful bacteria, but its neighbor, it's a very dangerous bacteria, just by sitting side by side, they can exchange this information, and the resistance can be delivered from the harmful bacteria to the pathogenic bacteria. So this is a concern.
Is this how resistance to antibiotics was created in the first place? Generally this herd immunity, as it were?
So there are many different mechanisms of spreading antibiotic resistance. Some of them are by mutation, so bacteria can accumulate mutation that can resist to this antibiotic. But maybe the most important contributor to the spread of antibiotic resistance are plasmids. And plasmid can be delivered between bacteria very fast.
What are plasmids?
Plasmid are like small circle DNA molecules. They exist as separate units that can spread by themselves in between the bacteria. And
So separate from the
chromosome. So they are like kind of like satellites that are associated with the bacterial chromosome, but they can be immediately transferred in between the bacteria. And they are like large plasmid that encodes their own delivery systems. They call conjugative plasmid, but also they are like small plasmid that can very, very rapidly exchange in between the bacteria in many different pathways. And one of these pathways are nanotubes. And we think it's a very common pathway that exists among many different bacteria, and even in this exchange can occur in an interspecies manner. So between different bacteria belonging to different species. So this is why we think it's a very important mechanism. And for us, it's just fascinating to see how bacteria are approaching each other and interacting with each other. And we're interested in understanding what are the cues for this interaction, what establishes this interaction, how do they choose their partner, and as you said, like what is going through this junction and what controls this flow of information, molecular information.
In Professor Ben-Yehuda's lab, they work with benign soil bacterium to explore these phenomena safely. Even though the strain they study isn't pathogenic, it shares traits with harmful bacteria, including the ability to form hardy spores and these intercellular connections. You said that bacteria today were very different from what they were 100 or 200 years ago. Can you elaborate on this and say, in what ways?
So I think mainly because of the antibiotic spread and the use of antibiotics everywhere, both in medicine, but also in agriculture, and like it just spreads. And this, I think, is the major driver for like obtaining these bacteria that are multi-resistant to many antibiotics, and it's risking our future because at the moment we don't have any good solution for like the era of the after antibiotics. So
is it around the corner? How far down the line is it?
It is around the corner. I'm not a specialist in like evaluating that, but there is a concern all over the world about this issue.
And if we follow your research and understand the mechanisms of communication between bacteria, what can it do to overcome resistance to antibiotics?
So our research helped to understand how antibiotic is spread and what are the factors within the bacteria that control this spread. So we even have like protein, for example, that blocks this spread that the bacteria is naturally producing. So if we understand these pathways of molecular exchange within bacterial communities in detail, in molecular detail, I think we will be more intelligent, we will be more prone to have innovative ideas how to block this spread and how to control this spread. And of course, we need all the time to develop new drugs. And this is also something that we have recently, based on this study, are attempting to do.
So it's gone already to a clinical stage?
No. It's a very, a very, very beginning. So it's just an idea that came from all our experiments. So some idea how to basically deliver a new type of antibiotics into bacteria, but still working.
Ben-Yehuda's discovery of how bacteria communicate offers a window into the hidden complexities of microbial life. But what about bacteria that choose silence, quietly defying antibiotics by simply going dormant? That's coming up next. But first, a word from our sponsors.
The view from Mount Scopus is made possible by American Friends of the Hebrew University. Established in 1925, AFHU has provided over a century of partnership and support to Israel's leading institution of higher learning. To learn more, visit afhu.org.
To unravel the mystery of bacterial persistence, we cross disciplines from microbiology to physics, meeting Professor Natalie Balaban and her interdisciplinary team. I head across town to meet Professor Balaban and her colleagues, Dr. Orit Geffen and Dr. Maskit Bar Meir, at the Rakach Institute of Physics. Equations are Professor Balaban's bread and butter, yet she's every bit as focused on bacteria as the microbiologists. Her interdisciplinary team is trying to answer a question that straddles biology and physics. I'll let her introduce the conundrum.
My name is Natalie Balaban, and I'm heading the lab of self-replicating matter in the physics department at the Hebrew University. The main interest of our lab is actually focused on understanding why one bacterium differs from an identical similar bacterium, especially in their response to antibiotic treatment. For some reason, even if you have twin bacteria, they are identical twins, one of these bacteria may die under antibiotic treatment while the identical twin may survive. And the big question is why? And we have a biophysical approach to this question.
How does a physicist approach an issue that is, about biology and bacteria, etc.?
I think what physicists are looking for in any system that they study is really to understand the core fundamental issue that is in front of their eyes. And sometimes it takes them to things that are completely irrelevant to the clinical world. We were lucky that this very basic question that we asked actually had also clinical relevance.
And what is the basic question in physical terms?
In physical terms, the basic question is why, when you have identical cells, and it could be any cells, could be also cancer cells, right? But we work on bacteria because it's easier. And the fundamental question is why identical cells that have the same DNA, twins, identical twins, at the level of bacteria are still behaving differently, and can we predict these differences? Can we really understand where they come from and what is their relevance for the clinical world, for example?
In addition to her work as a researcher, Dr. Maskit Bar Meir is a pediatrician at Shaarei Tzedek Medical Center in Jerusalem, where she specializes in pediatric infectious diseases. I was curious to hear how research meets practice in her daily life. How do you find it relevant for the clinical world? I mean, do you see, I'm guessing that resistance to antibiotics is a major issue for medicine today.
Except for resistance, I think the problem is our difficulties in treating resistant infections. So there's a difference between resistant bacteria and resistant infections. So the bacteria can be sensitive to the antibiotics, but still the infection itself is resistant to our treatment. And here it's really relevant. So
sorry, hang on a second. If the bacteria is not resistant to antibiotics,
so why the infection is resistant?
Exactly. Where does the infection come from?
Because that's the different behavior that Professor Balaban mentioned. Because the bacteria can behave differently in different areas of the body and in the lab. So when I have a patient with infection and I take the culture, take it out from his blood, for example, and take it into the lab, the bacteria has changed completely. So all the information I can give the clinician is completely different, or maybe completely different, from what happens in the body. And here is the gap that we're trying to close between what we call in vitro, which is the lab, and in vivo, the patient body or the area of the infection.
How wide is the gap between in vitro and in vivo?
I think it's huge, and we probably don't evaluate it as it. I mean, we underestimate it. For sure, we underestimate it, because we know that if we don't have an effective immune system, so for example, in patients who are immune compromised, have a sick immune system, then our ability to treat effectively the infection is our treatment is very limited, right? So for sure, and the bacteria is sensitive. In the lab, still the patient doesn't recover. So for sure, the gap is huge.
An important step towards closing that gap is first and foremost to identify and classify the different bacteria. Once a catalogue of the different pathogens is created, it is much easier to come up with an appropriate treatment. Whether it will be effective under different circumstances is a separate issue. But first, the clinicians need to know what they're up against. This is where Dr. Orit Geffen comes in. She started off as a PhD student, but now runs day-to-day research at the lab. Where does your role as an articulating link between the lab and the sickbed come into play?
One of the things that we did in the last years was the TD test, which is a method that help us to better identify bacteria in the clinic. There we started the connection between the clinic and the lab. And since then, we're working on collecting bacteria from the clinic, trying to identify them in the lab, and we're trying to do in the lab something that is as much close to in vivo than was ever done, I think.
And Musket, you are in charge of collecting the bacteria from the field, as it were. Can you describe how the process is done?
Actually, it changed over time as our cooperation progressed, because at first we collected in the regular way, which means we take cultures from the lab. So the patient is sick, the physician collects the bacteria is being processed in the lab, and then we just took it and examined the bacteria. But then we thought that we're collecting a bacteria that has changed already, dramatically changed. During the day or two in the lab, it's a completely different creature than it was in the patient. So we figured out that maybe if we take it straight from, let's say, from the blood, and freeze it immediately, fix it, and not letting it change at all, then we might get some more information that is as close as can be to in vivo, to what happens in the patient's blood.
I want to go back to the what you said is the fundamental question. Do you have any hypothesis as to why it is that bacteria that are so similar react differently?
Right, so just to make things clear, when you have a bacterium that becomes resistant with a mutation, so definitely it's different, it's not an identical twin anymore, and it's very clear why it survived antibiotic treatment. What we saw is that the viability of identical twins makes them not become really resistant to the antibiotics, so they are not able to grow in the presence of the antibiotics, but they are just able to survive. And this is one of the main reasons, is that sometimes some of these identical twins go randomly to sleep, right? And then you have one sleepy bacterium and one actively growing bacterium. And under antibiotics, they will react very, very differently. But this is transient, right? So in order to catch these differences in their physiology, you really have to look at them under the microscope. You have to develop specific tests that Ori talked about, the TD test. And what we are trying to understand is how these differences in physiology are actually at work in the patient.
Can you describe how they behave differently under the microscope?
I think the main difference is that the persistent bacteria are in like a sleepy mode, like they're dormant, and they remain dormant during the antibiotic period. If they wake up after the antibiotic treatment ends, they will survive it, because most of the antibiotic affect the growing system of the bacteria. So the normal bacteria will try to grow, and then the antibiotic will kill it, and the persistent bacteria will remain dormant.
Is dormancy the mechanism that allows bacteria to become resistant to antibiotics, or is it just one out of many mechanisms?
So it's not really resistance. We call it tolerance, just to differentiate it from resistance. But yes, the outcome is that you have an antibiotics, you take away the antibiotics, and then they are able to recreate the infection. So they contribute to treatment failure. We think it's a major way to contribute to treatment failure. Definitely in vitro, we see it all the time in any type of bacteria that people have tried to study. They are always there, these sleepy bacteria. But now we understand that there are many ways of being sleepy. And this is where actually the statistical physics helps us really describe the distribution of sleepiness on the population of bacteria, and it allows us also to predict how often we'll see these phenomena.
And is there a way to imitate like a living tissue in the lab or something like that?
Today in the lab, all we know to do is to anticipate resistance or sensitivity. But we don't have all this information, which is basically what happens to the bacteria over time, what it behaves like. It's like having a stills image of someone and having a video image of someone. So it's a huge gap.
Dr. Bar-Meir notes one finding already changing perspectives: the realization that more drugs are not always better. There's a tendency to think that combining two antibiotics will always increase the chances of success, but the persistence phenomenon complicates that.
For example, our work showed that drug combinations are something that you have to really examine. When you give two drugs, it's not necessarily better than one drug. Sometimes it's worse. So the awareness to drug combinations, to treating what we call biofilms, so bacteria that are that stick to foreign bodies, and the awareness to how much antibiotics you give, for how long, and which combinations is something that we try to promote. And the fact that the information from the lab is very limited. So the fact that we know that we have limitations in our knowledge is a big step.
How do you envisage treatment in the future? Once, say, like tomorrow morning, you clinch that holy grail, and you understand exactly how bacteria react to antibiotics. How do you think that clinical treatment would look like?
If I'm allowed to dream, then I think what we're trying to recreate in the lab are actually tests that are predictive for how the bacteria will behave in patients. So if we can devise a battery of tests that will tell us, this bacterium in the patient, it has this degree of sleepiness, this degree of resistance, which is something that is already measured, and taking all this data together, and it's in this niche in the patient, then by having reproduced these physiologies in the lab and manage to understand what combination of treatment would work, then we hope to actually reduce significantly the duration of treatment, the failure of treatment.
Yeah, so probably instead of just examining the resistance or the susceptibility with like discs of antibiotics, we probably will be able to take the bacteria out of the patient, immediately sequence it, the RNA sequence or the proteomics or whatever, characterize it in a much more sophisticated way and come up with these proteins are being produced, so then this kind of antibiotics would work or this one would not work.
From bacteria that persist despite antibiotics, let's rewind nearly a century, when the battle wasn't about drugs, but about swamps, mosquitoes, and the relentless spread of malaria. Professor Israel Kligler didn't have antibiotics at his disposal, only determination, strategy, and the power of community. While Saul Adler fought tropical diseases in the lab, Professor Israel Jacob Kligler tackled a public health menace in the field. Born and trained in the United States, Kligler arrived in British Mandate Palestine in the early 1920s to confront malaria, a disease then so rampant it threatened to derail the entire Zionist project. Swamps and marshes across the country bred malaria-carrying mosquitoes. Thousands fell ill or died every year. Kligler set out to eradicate malaria, an idea that sounded crazy at the time. I reached out to the preeminent expert on Kligler, a retired British lawyer and independent researcher named Anton Alexander, to learn more about him. I asked him just how central Kligler was to the story of malaria in the Holy Land.
Absolutely central. Absolutely. People don't realize that before Kligler came along, the notion of malaria control or malaria elimination just hadn't been considered anywhere, really. He comes along just after the First World War. The British mandate had said, well, we're not going to be involved in malaria control because it's just too costly. By the way, the Palestine front against the Turkish army was the only successful malaria front in the World War I. There were other malaria fronts, but they just wiped out the troops.
What does it mean, malaria front? Is it like a battlefield that is infested with malaria?
No, if there were countries in which there was malaria and the two armies faced up to one another, neither side could do anything because the troops would just go down with malaria. The big front that was badly affected was Macedonia. And I think on one occasion they called off a battle because they didn't have troops to fight.
When Kligler arrived in Mandatory Palestine in the early 1920s, the British authorities were skeptical about fighting malaria. It seemed too costly, too difficult, a lost cause. But Kligler was undeterred. He launched an all-out war on mosquitoes and their breeding grounds.
After the First World War, Kligler comes along about 2 or 3 years later. And for that reason, you're in Israel now because of Kligler, because he managed to control it so long that it was eliminated eventually. The first start of malaria elimination took place in Palestine.
Anywhere in the world?
Yeah. The principal object was not insecticide or bed nets or anything like that. They just got rid of the mosquito breeding sites. That was the main thing.
So it was more about behavior rather than science.
Well, they needed the entomologists to begin with to actually identify where the breeding sites were.
And that was Kligler.
That was Kligler, yes. But he needed the inhabitants to make sure that the breeding sites remained destroyed. And that was a big, big thing.
So he had a whole team working with him?
He mainly used the inhabitants. He encouraged them. He educated them.
Anton is adamant that without the eradication of malaria, there would be no state of Israel today. Can you say why?
Well, malaria is one of the big killers of the world. Or it debilitates. It just reduces a person's physical abilities to get on with things. Look, Palestine was a desolate country because of it. Remember, Petach Tikva had to be abandoned at one stage. Did you know that?
Yeah. So what they did was essentially allowing people to work and essentially build a country. Today, there's another epidemic raging, one deeply tied to behavior, environment, and reward. This time, it's not mosquitoes, but something even closer to home, the food on our tables. To learn more about the massive threat of obesity, I visited the laboratory of Professor Jonathan Kupczyk at the Faculty of Medicine.
My name is Professor Yoni Kupczyk. I'm the head of the Department of Medical Neurobiology, and my laboratory investigates the neural mechanisms underlying motivational pathologies like drug addiction and overeating and obesity.
What is the connection between the two, drug addiction and obesity? How do you see them as belonging to the same group?
Well, obesity is a condition that developed really in the last 50 years or so, and it was often regarded as a problem of metabolic or energetic balance. But we think, and you can see it also in studies, in human studies, that the problem is not a problem of energy in the body, but it's a problem of consuming, overconsumption of food, of highly palatable, highly processed food. And since this food is highly rewarding and it is designed to be highly rewarding, we think that the problem here is a problem of consumption of reward, and addiction is the same thing. Addiction is a problem of consuming rewards, and therefore we believe, or we think, that some of the neural mechanisms that may underlie overeating and obesity may be embedded in circuits in the brain that guide us to consume rewards.
Why are some people more prone to it than others?
That's the billion-dollar questions. We don't know yet. That's the true answer. We are looking for it. Also, in our research, we try to compare between animals that gain more weight than others under the same conditions, trying to see if physiologically they have different activity in the neurons of the reward system. So we did find that in animals that we call them obesity prone, so those are the animals that, given the same high-fat, high-sugar diet, gain more weight than others, we found in them neural activity that is similar to what we see in animal models of drug addiction. For example, in an animal model of addiction to drugs, we usually give animals the drug for a certain amount of time, and then we put them off the drug to model withdrawal. And then we examine the brain, the reward system, and we see overactivation of certain pathways. When we take animals, give them high-fat, high-sugar diet, and then put them off the diet to model, it's not abstinence from food, but it's going on a diet. And we see the same changes occurring in the reward circuits.
To better understand these similarities, I spoke with Shani Gendelis, a PhD student in Kupczyk's lab, who is investigating the differences between obesity-prone and obesity-resistant mice.
Hi, hello, my name is Shani. I'm close to finish my PhD in Yoni's lab. And my project is trying to identify difference in the physiology between those obesity-prone and resistant mice in specific brain areas that are part of the reward system. What we saw between these mice, the way those area, the reward area in the brain works, is that they put a continuous brake on consumption. And what we saw is that the mice that gained the most amount of weight, this brake weakened. And it caused the mice to consume more food.
The lab is also looking at how bariatric surgery might affect this reward system. Another student, Yair Lalouche, who is pursuing a combined MD-PhD, explains.
My project focuses on the effects of bariatric surgeries on the said reward system that we just discussed. So I'm coming from a different angle, from the therapeutic angle, and trying to see similar parameters both in the brain reward system physiology, how it's affected by those surgeries.
A common misconception about bariatric surgery is that it simply makes the stomach smaller, forcing people to eat less. But as Lalouche explains, the reality is much more complex.
Either more averse or more motivated to achieve healthier foods, like you see a shift to more vegetables, less fats and less sugars. And also in the behavioral level, we have tests to see what is the motivation, or it's on the border of, let's say, addiction, to achieve those kind of highly rewarding food and highly unhealthy food. And you see this motivation decreased significantly.
Professor Kupchik is a member of the Israel Center for Addiction and Mental Health, and his research raises profound questions about why we eat more than we need.
Is obesity a form of addiction? Our results do confirm that overeating of highly palatable food is driven by the reward system. The changes in the reward system that we see that happen in animals that overeat are similar to the physiology we see in the brain of animals that use drugs. I'm not sure yet if clinically we can say that obesity is a result of an addiction. This needs more research, but biologically it does seem as an addiction or overconsumption of reward. My goal is to understand what drives overeating, why, and this is the basic question that I still don't really understand. Our brain has developed over the years to really fine-tune everything we do. Consumption of water, all sorts of behaviors, body temperature, everything is controlled to be very, very accurate. And so is energy consumption. And yet, we eat more than we need. I mean, we all, not all, but most of us have dessert after a full meal, and we don't need it. And so something else overrides our energetic needs. And I think that's the reward part, the craving for the reward. And so if the reward system of some people works strong enough and consistently to drive us eat more food, more high-fat, high-sugar food, it would be similar to a reward system that drives people to consume alcohol, to consume nicotine, cocaine, heroin, all kinds of other rewards. So in that aspect, it does show similarity to other forms of addiction. Clinically, does obesity need to be treated by psychiatrists or by other doctors? I'm not sure. That's not my job to determine, but definitely overeating in the modern world of all these highly processed foods involves the overactivity of the reward system in a similar way that we see in addiction.
We need to remember it's almost 30-40% of the population that develop these kind of changes in the brain. So we need to think if it's really a condition or it's just part of the population. And if we look at it that way, then we need to see how we can look at these people. Because if you categorize them because of all the thought about psychiatry and the stigma about psychiatry, you need to think, because it's 30%, it's third of the population that develop these kind of conditions.
I would like to add, you asked an important question, I think, about the redefining of obesity as a psychiatric disorder. I think the more accurate way to look at it is obesity, as I see it, is some kind of triangle that is, or the reasons to obesity. There are, you have metabolism, how the body processes and the foods. You have the behavioral aspect, which we talked about today, which is actually the consumption of food, and you have the energy expenditure, which might be the physical activity, and also related to metabolism. So I think the treatment of obesity was until now mainly focused on metabolism, and the behavioral aspect of that was known, but was not really, the mechanisms were unknown, and therefore as a therapeutic direction it wasn't that strong. And I think maybe the future of obesity, now that we understand that the behavioral aspect has a great effect on this issue, is to go in a more multidisciplinary manner. Not meaning that the psychiatrists are going to treat it, but yes, the psychological aspect of it must be taken into account when you're treating your patient in a more holistic way that would bring more results.
I agree.
Well,
okay, we are in agreement. No, I just wanted to add that all this research on the crosstalk between the stomach or the digestive system and the brain is something that over the last 10, 15 years, people started to understand that there's a lot of crosstalk that could affect both ways. It's known for a long time that the brain controls, or the nervous system controls the digestive system. But now we have proof, more and more proof, that the digestive system can also control the nervous system and affect it.
From Adler and Kligler's heroic battles against tropical diseases to today's scientists facing modern health threats, antibiotic resistance, bacterial persistence, and obesity, the Hebrew University's story remains one of bold ideas and relentless inquiry. Whether it's decoding silent bacterial conversations or exploring the brain's response to rewarding food, these discoveries are more than just academic. They shape how we understand our bodies, our behaviors, and our futures. And that's all we have time for today. Thank you for listening to The View from Mount Scopus. Many thanks to executive producer Talka Oz, technical producer Leo Peleg, to Ruth Shaw, Luciana Hosanna, and the Hebrew University's Division for Advancement and External Relations and its American friends. If you liked what you heard, don't forget to subscribe and give us a 5-star rating. It really does help. And if you know someone who'd love this episode, go ahead and share it with them. In our next episode, we'll explore other facets of the Hebrew University's impact. We hope you'll join us again. And until then, shalom from Jerusalem, and see you next time on The View from Mount Scopus.